Good day everyone. Welcome to the week 7 of the course Biological Data Analysis and Visualization with R. So far we have talked about regression models in the last week. We'll now go a bit further, and in this week we'll be talking about something called dimensionality reduction. So I'll start with what is the importance of dimensionality reduction in biological data. So, and this is a very common tool that we apply to many biological data sets that are quite big now. And then we'll talk about dimensionality reduction techniques, right? Some of the most popular techniques that are used for this purpose. And then we'll also apply some of these techniques to our data in the hands-on part. So let's start with dimensionality reduction. What do we mean by dimensionality reduction? So when you look at modern biological data sets, they often have a very large number of dimensions. Okay, so we can take some example. For example, we have gene expression data sets. Depending on the organism, we often have data for 20,000, 30,000 genes per sample, and then of course on top of that we have multiple samples, right? So if you consider this data set, this becomes a really big one, right? We have 20,000, 30,000 gene data points for every sample, and then if we have 10 samples, that already becomes quite a big data. If you consider now single cell RNA sequencing gene expression data sets, so these are even larger, right? So we have a large number of cells and large number of genes. So we are looking at a very high dimensional data, right? So when you talk about dimension, right, these are the dimensions, right? We have 20,000 dimensions or 30,000 dimensions in here, okay? And on top of that, the number of samples will simply kind of make the data huge, right? Now, one of the things that we also realize, right, when you look into the expression levels of 20,000 genes in a sample, is that not all of these genes are important for determining a specific phenotype that we are studying, right? So let's say we are studying, right, how disease samples differ from normal samples, right? And we have done, let's say, gene expression profiling. And what we see is like only probably some 10%, 20% genes that show difference in their expression pattern, right? So as we now understand, out of the 20,000, probably 10% genes are very important, right? That actually kind of determine the difference between a disease sample and the healthy sample, right? So this is something that we can utilize, and we can see, right? Can we identify these most important features, right? And maybe not look at 20,000, 30,000 expressions. Maybe we can look into only expression levels of a few genes, and that is something would be enough to actually understand the difference. Okay, so this is where the dimensionality reduction comes in, right? So dimensionality reduction enables us to identify the most important features, at least some of the techniques. So using these different techniques, we can identify what are the most important features that actually differentiate these different samples. Okay, so this is something that is possible. The other thing that we can also do is dimensionality reduction also reveals similarity between samples. Okay, so when we are doing this dimensionality reduction, we take all samples. In this example, as I said, let's say we have 20,000 genes and we have, let's say, 10 samples. When you do dimensionality reduction, it actually also reveals that what are the similarities between samples, or which samples are similar to each other in terms of expression pattern. Okay, so expression pattern of all genes or the most important genes, and which samples are similar to each other and which samples are different. So the samples that are similar to each other in terms of expression pattern, they tend to form clusters, right? They tend to come close, right? And that sometimes lead to formation of clusters. Right, so if you can see, for example, that we have these clusters, right, different samples. So we have healthy cluster and disease cluster, right. So in that case, we can probably now start identifying the features that actually leads to the separation of these samples or separation of these clusters. So this is something that is very powerful in terms of looking into the data or exploring the data, right, and kind of understanding how the data set looks like. So the goal of all dimensionality reduction techniques is, like, of course, reduce dimensions and identify the most important dimensions, but also minimize loss of information. Okay, as you realize, if we start looking at samples as expression levels of only 100 genes, the most important 100 genes versus the 20,000 genes, of course, we are going to lose some information, right. We are going to lose information about some of the details of other genes that also show some variation between some samples, but not across all samples, right. So, when we reduce dimension, we will always lose information, but the goal when you apply these dimensionality reduction techniques, the goal is to minimize that loss of information. Okay. So, we want to reduce dimension, but we want to keep as much information as possible. Okay. And this is what these dimensionality reduction techniques do. Okay. So, with that introduction, we now understand that dimensionality reduction techniques are very useful for exploring biological data sets, for looking at biological data sets, and sometimes it can give us valuable insights into the what is actually happening in a process. So, we apply these techniques right across gene expression data sets when we are working with single cell RNA sequencing data sets, where we typically have very large dimensions, as I mentioned. But we can also apply this to any data sets where we have multiple sets of features, and we want to understand how these features contribute to the final outcome. Okay. So, this is something that we can visualize and explore with using dimensionality reduction techniques. What is important to know at this point is that these techniques, they cannot do something like regression or prediction that we talked about in linear regression, right. So, this is something that we cannot do. What these techniques can do, they can give us the structure of the data, right, or how much variation there is in the data and which samples vary from each other, which samples are similar. That kind of structure we can understand, we can explore, but these methods will not give us any prediction like we did in linear regression. Okay. So, this is something that is very important to remember. So, with that background, let us now move on to the dimensionality reduction techniques. So, we will briefly discuss two types of dimensionality reduction techniques. The first type is the linear dimensionality reduction techniques, such as principal component analysis, a very popular type of dimensionality reduction techniques. In short, it is called PCA. So, what happens when you do dimensionality reduction using PCA? The key features are extracted, and these are represented as something called principal components. Okay. So, when you do principal component analysis and dimensionality reduction using this method, what we end up with is usually a list of principal components, right. So, starting from principal component 1, 2, 3, 4 and so on, right. You can have a very large number of principal components, but it is usually the first few principal components that contain the most information. We will learn about this as we go into the hands-on part. So, you will see how these PCs are informative about the data. What these principal components contain are, so these are actually weighted combinations of original variables. Okay. So, let us say we are working with a very big data set where we have 20,000 gene expression for 10 samples. At the end of a principal component analysis, what we will end up with, let us say we will get a list of PCs, right, principal components for, and then we have the values for each sample with those for those principal components. Now, what these PCs are? These are simply weighted combination of the features or the genes that we have. Okay. So, this is what we get, right. The PCs are weighted combinations of original variables. Now, these combinations are linear combinations, right. So, that is why it is called linear dimensionality reduction technique. Okay. So, this is the PC1, for example, could be a weighted combination of, let us say, x1, x2 and so on. Okay. So, we can take this example of gene expression data where I said, right, we have, let us say, 20,000 to 30,000 genes, and we want to extract most important features as PCs, right. So, these PCs, right, PC1, PC2, these will be the weighted combination of genes, and this is something what we refer to as principal component loadings. Okay. And we look into these PC loadings in our hands-on analysis. Okay. So, I hope we understand this clearly now. So, instead of the original variables, right, original features which are the genes in this gene expression data set, we transform the data in such a way that we end up with something called principal components, which are linear combinations of the original features or genes, right. So, these principal components are calculated based on some weighted combinations of the original features. We'll call them as loadings, and this is what we'll see again how we actually can look into the loadings and what are the variables that actually have contributed to PC1, what are the variables that have contributed to PC2, and so on. What is also important to remember is that maybe only a few genes in this example will contribute to PC1, and maybe another set of genes will contribute to PC2, and a third set of genes will contribute to PC3. So, it is not always necessary that all genes, all 20,000 genes will contribute to each PC. Okay? This is something that we will see again, and this is called weighted combinations, because you will see one gene, maybe a specific gene, gene A will have larger weight, right? It will contribute the most towards, for example, PC1, and then you will have other genes that will contribute slightly lower than that, and so on. Okay? So, this is something again will be clear when you actually go into the hands-on part. So, as I said, right, these principal components are transformed. So, they are resulting from a transformation of the original data set, right. So, what we want to do is now we want to discuss these PCA steps, right. What are the steps that we perform to actually get these PCs, okay, very briefly without going into the full mathematics. So, what are the steps? The first step is data standardization. So, if you are working with the data where you have, let us say, 10 samples and 20,000 genes, the first thing we do is we standardize the data. When I say standardization, it is very similar to standard normal distribution, right. So, we have scaling of the data and we divide by the standard deviation, right. So, that is a data standardization step. So, this is something that is important, because if you do not do this, what will happen is the gene that has the highest expression is likely to influence the PCs the most. Okay. So, this is something we need to also keep in mind. Now, once we have standardized the data, what we do is we actually calculate the covariance matrix from the original data, right. So, this is covariance matrix between genes in our example of gene expression. So, covariance matrix for the correlation between the features, right, all the features that we have in the data, and it is important to remember that we are not distinguishing between the y and the x here, because as I said, there is no such predictive power of this model, right. So, there is no dependent variable, independent variable. PCA does not distinguish between all those, right. So, everything is taken and the covariance matrix is calculated. Once this covariance matrix is calculated, from this matrix eigenvectors and eigenvalues are calculated. Okay. So, we will not go into the details. There are some specific matrix processing steps that are there to actually get to the eigenvectors, and for each eigenvector there will be a corresponding eigenvalue. Now, we can sort these eigenvectors by eigenvalues, and these actually results in the principal components. Okay. So, these eigenvectors are the principal components of the data. Okay. So, as you can see, these eigenvectors will be combinations of the original features that we have in the data. Okay. So, very briefly, these are the steps. So, we understand what we are getting, right. We go through these steps and we convert these features into principal components. Now, what are the advantages of principal component analysis? And this is a very popular technique for specific reasons, and the first reason is this is interpretable. So, what I mean by interpretable is that we can identify the most important features in the data. Okay. So, this is something that this kind of technique allows us to do. We can identify the genes that are most important in terms of explaining the variation in the data. Okay. And it can also quantify importance of variables in explaining the variance in the data. So, one thing you will see is that the principal component is usually the most important component, right, for our analysis. That is the most important principal component, PC1, right. So, that is why as you have seen in the steps, we sort them by eigenvalues. So, the one with the largest eigenvalue is the first principal component, and that explains the most amount of variation in the data. So, the original features that actually contribute to this first principal component, they are likely to be the most important ones, and we can also quantify the importance of variables in explaining the data. We can also use this for feature selection, right. So, as I said, if we identify the variables that kind of explains the most of the variance in the data, we can choose these as features for building subsequent predictive models, right. So, this is something that is very useful if we want to apply such machine learning models on biological data, etc. So, this is something that's probably you understand. Now, what we will do in the hands-on part, we will do PCA and we will use 3 functions. One function is the PRComp. So, I will show you how this function works and how this function does PCA analysis. This is the built-in function in R for PRComp. Then we will use this R package factomine R. This is again a package that can do PCA analysis. Along with this, we also have a package that can help us visualizations of the results that we get from this package. There is a third package which we will also use, is called PCA tools, and this is something specifically built for analysis of gene expression data, but again it can be applied to data sets with many dimensions, so that it becomes very useful for us to look at the data. Okay, so this is something we will do in the hands-on part. So, we have now understood the linear dimensionality reduction technique, which is principal component analysis, and then we will also explore this further with the hands-on part. Now, before we go into the hands-on, I just wanted to very briefly mention there are non-linear dimensionality reduction techniques as well, and one of them is called uniform manifold approximation and projection, okay, or UMAP. Okay. Again, without going into the data, I just very briefly mentioned this is a graph layout algorithm for data representation. Now, what is important to know, again we are reducing dimensions, right. So, we have UMAP 1, UMAP 2, right, similar to PC1, PC2, but then these are not linear combinations of the features, right. So, which means these are actually this is not a very interpretable model, right. So, you cannot probably guess which features are important using this method. So, the goal of this method, right. So, as we are looking at, for example, gene expression data sets, and these methods are widely used for exploration of gene expression data sets, is that when you use these algorithms, right. So, their goal is to actually represent the data in low dimensional space, and this should closely resemble the data in the high dimensional space. Okay. So, we want to again preserve information as much as possible, although there would be loss, but we want to minimize that information loss here. Okay. So, this is something that is very important. Now, for UMAP analysis, I will just very briefly mention the key parameters that are important and we have to remember. So, for example, when you are doing this UMAP analysis, we will again do the hands-on. We will see that we have to consider number of neighbors considered for UMAP. There is a default value, but of course, we can choose whether we want lower value than that or we want higher value than that. Now, what happens is if you choose low values or here for this number of neighbors, you will kind of get the local structure in the data, right. So, you kind of maintain the local structure of the data. Whereas, if you choose high values, what you will get is mostly like the global structures. You will maintain the global structure in the data, and you kind of look into the global structure. So, again, depending on the application, one of them would be more or less useful for you. The other parameter, so the first parameter is number of neighbors considered for UMAP analysis. The second parameter is the something called minimum distance. Okay. So, what should be the minimum distance that should be considered for this neighboring, right. So, we consider two data points or samples as neighbors. Okay. So, if you again it is very similar to what we discussed, like number of neighbors. So, if you have large distance, you are maintaining the global structure, right. You can visualize the global structure, whereas if you choose minimum distance as like small, then you are probably preserving the local structure, right. So, this is something that we again have to keep in mind. You can try this in hands-on, we can vary these parameters and we can see how these structures will vary. So, what about how does UMAP compare against the principal component analysis? Okay. So, one of the drawbacks of course of UMAP is this is less interpretable. As I said, when you reduce the dimensions, UMAP 1, 2 or 3, right, these are not combinations, these are some combinations, but these are not interpretable, right. So, again unlike PCA, right, here there is no linear combination or non-linear combination that is there in the UMAP 1 or UMAP 2. So, we do not know which features are actually the most informative ones by just doing UMAP. The other thing here is very important is the distance between clusters in UMAP. This is not important, okay, and this is again we need to remember. So, in PCA these clusters we will see and the distance means something. So, distance kind of measuring the expression pattern. Here in UMAP this does not mean anything, okay. So, we cannot do any analysis between the distance between clusters. So, in linear dimensionality reduction techniques, if we have like if the distance is high, right, which means their clusters are dissimilar, here the distance does not mean anything. Here again UMAP the cluster size does not provide any important insight, okay. So, this is again we need to remember as you can see that the cluster size might vary depending on the parameters that we choose. And finally, of course, we have no idea about the contribution of the features that we are going to work with, right. So, which features contribute to this clustering, this is not clear by just doing this UMAP analysis. Whereas, if you remember in PCA we kind of understand, right, how these features are important. So, the hands on part, right, which will do in UMAP is the package called R package UMAP, okay. So, this is something again that will come in the hands on part, okay. So, with that let us now move on to the PCA hands on first, and then we will follow that up with the UMAP hands on, okay. So, let us now go towards the first PCA analysis with the PRCom package, and we will see how this works. Okay, so, I will just introduce the data. So, let us see where we are. Okay, so, we have to move to the right directory first. Okay, so, this is where our data set is, right, and there will be two data sets that we will be working with, and as we have seen, the first one is called PCA cancer gene expression data. This is what the first one, and then we will be working with the yeast gene expression noise data. Okay, so, we will apply different tools in these cases and we will see the data. Okay. So, the first step is the as we said, right, we will first work with the PRComp data, right. This is the principal component, that is a built-in function in R, and we will first work with that, and then in the next class we will be talking about the Factomine package, right. So, Factomine R we will be talking about that package. Okay. So, let us first look into the data, right, and let us load the data. So, the data file is here 100 genes PCA2 new .csv. Okay, this is the file, and in addition what we have is the data labels. Okay, data labels they contain label for the data. So, I will see in a moment, I will show you in a moment how this data looks like. So, okay, so we need this library dplyr, right. This is for data organization, and let us load the original data. This is in CSV format. So, we need to use this command read.csv. Okay. All right. So, this is loaded now. Let us look into the data. Okay. And what you notice probably, and I can also type it for you, is that these are gene names, right. So, these are gene names, about 100 gene names, right, or slightly more. You have 103 genes here, right, for which we have expression data for multiple samples. Now, the sample names are not given, but their label is given, right. So, you see this last column, there is something called label. Okay. So, this label actually describes which sample does it belong to. Okay. So, we will see this in the labels file. So, we will read this labels file now. Okay. And what you will simply see here is data labels, Okay. So, here data labels, what we see, the first column is saying type, the second column is abbreviation, and the third column is the label. Okay. So, this label value is 0, 1, 2, etc., and the range is up to 31, you will see 30 or 31. So, you have 31 classes of data. What each of these label is referring to is a cancer type. Okay. So, here you see the type, cancer type that is mentioned, and the abbreviation, that is the short name for each cancer type. So, for example, here we have acute myeloid leukemia, right, LAML, we have adenocortical carcinoma, ACC, bladder urothelial carcinoma. So, all these cancer types that are actually labeled here. So, we have 31, so 0 to 30, as you can see here, right. So, up to 0 to 30, and for each cancer type we have a specific abbreviation, and the description is here, right, which cancer type we are talking about. Okay. So, this label now gives us which type of cancer this sample belongs to. Okay. And this is what we can see in the original data. Okay. So, as you can see here, the label is 0, means this is one cancer type, and then we can also see the tail, right, and they are, because they are organized. So, you will see the last few samples belong to 30, right. So, they are kind of ordered now from label 0 to label 30. Okay. So, we are looking at a large number of samples. We can also look into the dimension of this original data. So, it is a pretty big data set where we have 10,000 samples and expression for 103 genes, and we also have the label. Okay. So, this is something is actually good for principal component analysis, and we can see, okay, whether the cancer samples actually clustered together, right, the samples that belong to the same cancer type, whether they clustered together, and can we identify the features that are actually the most important features for separating out these clusters, right. So, these genes, as you can realize, these genes are likely to explain the difference between different cancer types, right. They are likely to be able to give us some insights into the biological process. Okay. So, we can also look into this, right. So, we can do something called unique original data labels. So, it will tell us, right, what are the labels, right, different labels that we have which are unique, right, not the, if you have multiple zeros, it will just report 0 ones. If you have multiple 1 labels, it will report 1 ones, right. So, that is why the unique command we have, these are the labels 0 to 30, these are different cancer types that we have in the data set. Now, what we will do is, we can also do this, right, is something called original data group by label. We have seen this in case when we are talking about DPL-YR package data organization, right. So, we will do this group by label, and then we can count. So, what we will get at the end here is we will get the number of samples for each cancer type, right. So, for each level means for each cancer type. So, we will taking this original data group by label and count level, right. Count level, we have seen this command again. This is a table. So, what we have is 0 to 30, this is the label here, and n, right, number of samples, this is the count that we have, and for each cancer type we have these many samples, right. So, for the first one, right, or the label 0 we have 173 samples, for label 1 we have 79 samples, for label 2 we have 426 samples, right, so on. So, up to level 30. Okay. So, we know how many samples we have for each cancer type. Okay. This is something that we can see here. Now, what I am going to do next is I am going to choose only a few levels, okay, a few genes from only few samples from few levels. Okay. So, as I showed you, right, the dimension is 10,000, right, we have 10,000 samples and 100 genes. It is quite big data. So, if you have to do the PC analysis right now with all of this using PRComp or other functions, it might take a lot of time. So, I will show you this with only a small number of samples, but you will get the flavor, right, how you actually have to do this analysis, and then you can try with all 10,000 data sets and even bigger number of genes, right. So, if you have 10,000 samples, 20,000 genes, you can do this principal component analysis. Of course, that will take a lot of time and maybe a lot of memory. Okay. So, I will demonstrate this with a very small number of samples. So, that is why I am choosing only a few samples, and this is how I am going to choose. Okay, is data new, right, and from original data we have 1 to 20, we are choosing 1 to 20, 174 to 193, 253 to 272. Okay. And you might ask why these specific numbers, because as you can see from our data, right. So, first cancer type is 0 to 173. So, we are taking only first 20 samples, right, 1 to 20 for this one. So, we will have level 0 samples. Then we have 174 to 193. As you can see, after 173, right, we are taking 20 samples. So, from 174 to 20 samples for level 1, and then we are taking 20 samples for level 2. Okay. So, this is just to make sure that this analysis runs within a short period of time. But of course, the idea remains the same, right. If you want to run this on full data, you can simply run this here, saying, or like do the principal component analysis on the full data. Okay. So, you do not have to do this data new, etc. Okay. So, this is fine, we are taking these columns, right, and then what we are doing is we are taking again only first 20 genes. Okay. So, as you can see that this is a matrix form. So, these are choosing the rows, means the samples, and this is choosing the columns. Okay. And what this part is doing is choosing only first 20 columns. So, we are taking only 20 genes for our analysis. So, data for 20 genes only, as well as the label column, right. So, this is something we will need, because we want to actually look at this after the principal component analysis, if we want to see like whether different cancer samples are clustering together. Okay. So, that is what I am going to do, right. We are kind of taking a small data set, right, and we are also keeping the level, because that will be important for our subsequent analysis and visualization. Okay. So, here it is, and let us see, I can also do the dimension. Okay. So, we have 60 samples, 3 cancer types, and we have 20 genes, plus the last column is the level, right. So, that is what we have. As you can see, right, we have 20 genes, last column is level, and we have 60 samples. Okay. So, this is a small data set, we can run the principal component analysis much more comfortably. What we want to do also, we want to add these labels now. Okay. So, what we will do is we are creating these labels now and replacing these numbers, and we are using these abbreviations for cancer types. Okay. And data new $label is we are actually setting label values, instead of 0, 1, 2, we are now putting these. Okay. So, putting these actual cancer types. So, you will see these labels have now changed. We can check this by saying we have 3 cancer types, right, that we have chosen: LAML, SCC, and BLC. Okay. Okay. So, the last thing we will do is we will create this data set, and we just want to take those columns whose name does not match with label, right. So, because we are not going to do principal component analysis with the label column, right. So, we just choose the columns where we have information for genes, okay, and we kind of discard the label columns for the principal component analysis. Okay. So, I think I will stop with that in this class. We have looked into the initial steps, and we will continue this in the next class. So, I will go back to the presentation. So, just to summarize, as I said, dimensionality reduction is necessary for extracting meaningful insights. As we have seen, we can identify some of the important features, which we will do in the subsequent classes in the hands on, and this can also give us the structure of the data and similarity between different samples. There are different types of dimensionality reduction techniques. One I mentioned is the linear dimensionality reduction technique called principal component analysis, or PCA, and as I mentioned, PCA is interpretable, and it allows us to identify the key features that explain the variation in the data. And then we also have non-linear dimensionality reduction technique like UMAP. UMAP also allows us to see the structure in the data, but is less interpretable. So, what we will do in the next class is we will continue with the hands on, and we will see all of these with real data. Thank you.